Hello everyone. Today we are going to study about the remaining part of serological test. The first one we are going to see the immunodiffusion. So first is introduction. Immunodiffusion is a technique for studying the reaction between antigen and antibodies by observing participants formed by the combination of specific antigen and antibodies that have diffused in a gel in which they have been separately placed. Okay, so what happened in this also, see, you know that this all serological type, what we are going to observe, we are going to observe antigen antibody reaction. So in this also we are going to see, but with the help of gel in which they have been separately placed. Both in one gel they have kept the antigen, in one gel they have kept the antibody, and they are going to observe the reaction. So it has two categories. One is single radial immunodiffusion, one is double immunodiffusion. The first principles, how it works, how we know that. In authentic double diffusion, both antigen and antibody allow to diffuse into the gel. This assay is frequently used for comparing the different antigens preparation. The method called double, since the antigen and antibody are allowed to migrate towards each other in a gel, and a line of participation is formed where the two reactants are met. The participation is highly specific and used by the people working with the diagnosis and protein detection technique. See, if you see this picture, see, in one gel they have used the antigen, in one gel antibodies. Both is coming together and they are joining these participants. Okay. Next one is complement fixation test. So complement is a protein, that is globulin, present in the normal serum. Whole complement system is made up of 9 components, that is C1 to C9, and complement protein are heat level and are destroyed by heating at 56 degrees centigrade for 20 to 30 minutes in a process called heat inactivation. And complement binds to an antigen antibody complex. When the antigen is complexed and with antibody on surface of cell, complement causes lysis of cell. And in general, complement fixation tests are best performed in the reference laboratories where facilities exist for the careful standardization and control of reagent which this test required. And what is the principle for this? The complement fixation test is a technique that has been used over many years to detect and quantify the antibodies that serology does not agglutinate to or participate when reacted with its antigen, but can be demonstrated by its use or fixation or complement. Okay, the next is immunofluorescent test. Okay, first you have seen the immunodiffusion, then you have seen complement fixation, now is immunofluorescent. Antibodies can be labeled with the fluorescent dyes and can localize binding sites on cell. So here we are going to use the dyes. Okay, and the dyes is fluorescent, rhodamine, okay, phosphoerythrin can be conjugated to Fc reagent of antibody. So antigen binding is unaffected. Okay, and absorb at one wavelength and emit at another. Our next test is ELISA or ELIS. Okay, the ELISA is enzyme linked immunosorbent assay. E for enzyme, L for linked, I for immuno, S for sorbent, and A for assay. It can be used both qualitatively and quantitatively to measure antigen antibody binding. And depending on what variation you use, it will detect antigen for hormone, for enzyme, for microbial antigen, for illicit drugs, or antibody like anti-HIV in the screening test for HIV infection. So this ELISA test is usually you can use for HIV infection also, okay, to diagnose HIV, and in body fluids or tissue culture supernatants. Okay. So what it do? It's also called as ELISA. It also called as enzyme immunoassay, or it also called as EIS. Same thing only. Okay. ELISA is called as in a different name. Okay, is a biochemical technique used mainly in immunology to detect the presence of an antibody or an antigen in a sample. So it helps to diagnose the HIV. And the ELISA has been used as a diagnostic tool in the medicine and plant pathology as well as the quality control checking. So different method of ELISA. One is direct, one is indirect, one is sandwich, one is non-competitive ELISA. Okay, types of ELISA. Okay, direct, indirect, sandwich, and non-competitive. Okay. Uses of ELISA. It helps in the detection of antigen, antibody, hormones, and the enzyme. An example in microbiology, antigen HBsAg or antibodies HIV, HCV, CMV, and several other diseases. Next one is immunofluorescent. So the purpose of immunofluorescent is to detect the location and relative abundance of any protein for which you have an antibody. So once you have antibody to your favorite protein, you can use them to indicate where the protein is located. So this procedure is done to know the location of the protein. Okay. So this is the how they will do the test in the lab. Okay. Our next types is Western blot. So Western blot analysis can detect your protein of interest from a mixture of a great number of proteins. Western blotting can give you information about the size of your protein. So the last one we have seen, they will tell you where the protein is situated, okay, location of the protein. But this type will help you to tell the size of the protein, okay, with the comparison to a size marker or ladder in a KDA, okay, with the one marker they will put, no, with that they can compare it, okay. That's why the name came Western blot, and also give you the information of protein expression with a comparison to a control such as untreated sample or another cell type or tissue. The method originated from the laboratory of George Stark at the Stanford. The name Western blot was given to this techniques by W. Neil Vernet and is a play on the name Southern blot, a technique for the DNA detection developed earlier by the Edwin Southern. The detection of RNA is termed Northern blotting. So this is how the Western blot name came. So with this we have finished all the types of serological test. You can go through it, and if you have any doubt you can ask. Thank you everyone.